Welcome to Flourish. I'm Diane Planidin, and you're in the right place if you're ready to create an inspired life. And we do so by working on our own personal development so we can be strong role models for those we love and mentor. Today is the final chapter in week 3 at Psych-100 in my Queen's University journey online learning. And it's all about the nervous system. So let's get started. The mammalian nervous system is a complex biological organ which enables many animals, including humans, to function in a coordinated fashion. The original design of this system is preserved across many animals through evolution. Thus, adaptive, physiological, and behavioral functions are similar across many animal species. Comparative study of physiological functioning in the nervous systems of different animals lends insight to their behavior and their mental processing, and making it easier for us to understand the human brain and behavior. In addition, studying the development of the nervous system in a growing human provides a wealth of information about the change in its form and behaviors that result from this change. The nervous system is divided into central and peripheral nervous system, and the two heavily interact with one another. The peripheral nervous system controls volition and nonvolition behaviors using cranial and spinal nerves. The central nervous system is divided into the forebrain, midbrain, and hindbrain, and each division performs a variety of tasks. For example, the cerebral cortex in the forebrain houses sensory, motor, and associative areas that gather sensory information, process information for perception and memory, and produce responses based on incoming and inherent information. To study the nervous system, a number of methods have evolved over time. These methods include examining brain lesions, microscopy, electrophysiology, electroencephalography, and many scanning technologies. The learning objectives: describe and understand the development of the nervous system, learn and understand the two important parts of the nervous system, explain the two systems in the peripheral nervous system, and what you know about the different regions and areas of the central nervous system. Learn and describe different techniques of studying the nervous system. Understand which of these techniques are important for cognitive neuroscientists, and describe the reason for studying different nervous systems in animals other than human beings. Explain what lessons we learn from evolutionary history of this organ. As previously mentioned, I am a student, I am not a teacher, and you are on a learning journey with me here today. This is open courseware, by the way. Evolution of the nervous system. Many scientists and thinkers believe that the human nervous system is the most complex machine known to man. Its complexity points to one undeniable fact: that it has evolved slowly over time from simpler forms. Evolution of the nervous system is intriguing not because we can marvel at this complicated biological structure, but it is fascinating because it inherits a lineage of long history of many less complex nervous systems, and it documents a record of adaptive behaviors observed in life forms other than humans. Thus, evolutionary study of the nervous system is important, and it is the first step in understanding its design, its workings, and its functional interface with the environment. The brain of some animals like apes, monkeys, and rodents are structurally similar to humans, while others are not. Does anatomical similarity of these brains suggest that behaviors that emerge in these species are also similar? Indeed, many animals display behaviors that are similar to humans. For example, apes use nonverbal communication signals with their hands and arms that resemble nonverbal forms of communication in humans. If we studied very simple behaviors like physiological responses made by individual neurons, then brain-based behaviors of invertebrates look very similar to humans, suggesting that from time immemorial, such basic behaviors have been conserved in the brains of many simple animal forms, and in fact are the foundation of more complex behaviors in animals that evolved later. Even at the microanatomical level, we note that individual neurons differ in complexity across animal species. Human neurons exhibit more intricate complexity than other animals. For example, neuronal processes, dendrites, in humans have many more branch points, branches, and spines. Complexity in the structure of the nervous system, both at the macro and micro levels, give rise to complex behaviors. We can observe similar movements of the limbs as in nonverbal communication in apes and humans, but the variety and intricacy of nonverbal behaviors using hands in humans surpasses apes. Deaf individuals who use American Sign Language express themselves in English nonverbally. They use this language with such fine gradation that many accents of ASL exist. Complexity of behavior with increasing complexity of the nervous system, especially the cerebral cortex, can be observed in the genus Homo. If we compare sophistication of material culture in Homo habilis 2 million years ago and Homo sapiens, the evidence shows that Homo habilis used crude stone tools compared with modern tools used by Homo sapiens to erect cities, develop written language, embark on space travel, and study her own self. All of this is due to increasing complexity of the nervous system. What has led to the complexity of the brain and nervous system through evolution? To its behavioral and cognitive refinement. Darwin proposed two forces of natural and sexual selection as work engines behind the change. He prophesied psychology will be based on a new foundation, that of necessary acquirement of each mental power and capacity by gradation, that is, psychology will be based on evolution. Development of the nervous system. Where the study of change in the nervous system over eons is immensely captivating, studying the change in the single brain during individual development is no less engaging. In many ways, the ontogeny development of the nervous system in an individual mimics the evolutionary advancement of this structure observed across many animal species. During development, the nervous tissue emerges from the ectoderm through the process of neural induction. This process causes the formation of the neural tube, which extends in a rostrocaudal plane. The tube, which is hollow, seams itself in the rostrocaudal direction. In some disease conditions, the neural tube does not closely caudulate and results in abnormality called spina bifida. In this pathological condition, the lumbar and sacral segments of the spinal cord are disrupted. As gestation progresses, the neural tube balloons up at the rostral end, and forebrain, midbrain, hindbrain, and the spinal cord can be visually delineated. About 50 days into gestation, 6 cephalic areas can be anatomically discerned. The progenitor cells, neuroblasts that form the lining neoepithelium of the neural tube, generate all the neurons and glial cells of the central nervous system. During early stages of this development, neuroblasts rapidly divide and specialize into many varieties of neurons and glial cells, but this proliferation of cells is not uniform along the neural tube. That is why we see the forebrain and the hindbrain expand into larger cephalic tissues than the midbrain. The neuroepithelium also generates a group of specialized cells that migrate outside the neural tube to form the neural crest. This structure gives rise to sensory and autonomic neurons in the peripheral nervous system. The mammalian nervous system is divided into central and peripheral nervous systems. The peripheral nervous system is divided into somatic and autonomic nervous systems, whereas the somatic nervous system consists of cranial nerves and spinal nerves and is under the volition control of the individual in maneuvering bodily muscles. The autonomic nervous system, also running through these nerves, lets the individual have little control over muscles and glands. Main divisions of the autonomic nervous system that control visceral structures are the sympathetic and parasympathetic nervous systems. At an appropriate cue, say fear-inducing object like a snake, the sympathetic division generally energizes many muscles, hearts and glands, adrenals, causing activity and release of hormones that lead the individual to negotiate the fear-causing snake with a fight or flight response. Whether the individual decides to fight the snake or run away from it, either action requires energy. In short, the sympathetic nervous system says, go, go, go. The parasympathetic nervous system, on the other hand, curtails undue energy mobilization into muscles and glands and modulates the response by saying, stop, stop, stop. This push-pull tandem system regulates the fight or flight response in all of us. The central nervous system is divided into a number of important parts, including the spinal cord, each specialized to perform a set of specific functions. Telencephalon, or cerebrum, is a newer development in the evolution of the mammalian nervous system. In humans, it is about the size of a large napkin, and when crumpled into the skull, it forms furrows called sulci, singular form sulcus. The bulges between the sulci are called gyri, singular form gyrus. The cortex is divided into two hemispheres, and each hemisphere is further divided into 4 lobes, which have specific functions. The division of these lobes is based on 2 delineating sulci. The central sulcus divides the hemisphere into frontal and parietal occipital lobes, and the lateral sulcus marks the temporal lobe, which lies below. Just in front of the central sulcus lies an area called the primary motor cortex, which connects to the muscles of the body and, on volition command, moves them. From mastication to movements in the genitalia, the body map is represented on this strip. Some body parts, like fingers, thumbs, and lips, occupy a greater representation on the strip than, say, the trunk. This disproportionate representation of the body on the primary motor cortex is called the magnification factor and is seen in other motor and sensory areas. At the lower end of the central sulcus, close to the lateral sulcus, lies the Broca's area in the left frontal lobe, which is involved with language production. Damage to this part of the brain led Pierre-Paul Broca, a French neuroscientist, in 1861 to document many different forms of aphasia in which his patients would lose the ability to speak or would retain partial speech impoverished in syntax and grammar. It is no wonder that others have found subvocal rehearsal and central excessive processes of working memory in this frontal lobe. Just behind the central gyrus in the parietal lobe lies the primary somatosensory cortex on the postcentral gyrus, which represents the whole body receiving inputs from the skin and muscles. The primary somatosensory cortex parallels, abuts, and connects heavily to the primary motor cortex and resembles it in terms of areas devoted to bodily representation. All spinal and some cranial nerves send sensory signals from skin and muscles to the primary somatosensory cortex, close to the lower end of the strip. Curved inside the parietal lobe is the taste area, which is involved with taste experiences that originate from the tongue and so forth. Just before the parietal lobe and under the caudal end of the lateral fissure in the temporal lobe lies the Wernicke's area. This area is involved with language comprehension and is connected to the Broca's area through the arcuate fasciculus nerve fiber that connects these 2 regions. Damage to the Wernicke's area results in many kinds of Agnosias. Agnosia is defined as an inability to know or understand language and speech-related behaviors. So an individual may show word deafness, which is an inability to recognize spoken language, or word blindness, which is an inability to recognize written or printed language. Close in proximity to the Wernicke's area is the primary auditory cortex, which is involved with audition. And finally, the brain region devoted to smell is tucked away inside the primary olfactory cortex. At the very back of the cerebral cortex lies the occipital lobe, housing the primary visual cortex. Optic nerves travel all the way to the thalamus and then to visual cortex, where images that are received on the retina are projected. In the past 50 to 60 years, visual sense and visual pathways have been studied extensively, and our understanding about them has increased manifold. We now understand that all objects form images on the retina are transduction in neural language handed down to the visual cortex for further processing. In the visual cortex, all attributes, features of the image, such as the color, texture, and orientation, are decomposed and processed by different visual cortical modules, and then recombined to give rise to singular perception of the image in question. If we cut the cerebral hemispheres in the middle, a new set of structures come into view. Many of these perform different functions vital to our being. For example, the limbic system contains a number of nuclei that process memory and attention and emotions. The globus pallidus is involved with motor movement and their coordination. The hypothalamus and thalamus are involved with drives, motivations, and trafficking of sensory and motor thought puts. The hypothalamus plays a key role in regulating endocrine hormones in conjunction with the pituitary gland that extends from the hypothalamus through a stalk. As we descend down the thalamus, the midbrain comes into view with superior and inferior colliculi, which process visual and auditory information, as does the substantia nigra, which is involved with notorious Parkinson's disease and the reticular formation regulating arousal, sleep, and temperature. A little lower, the hindbrain with the pons processes sensory and motor information employing the cranial nerves, works as a bridge that connects the cerebral cortex with the medulla, and reciprocally transfers information back and forth between the brain and the spinal cord. The medulla oblongata processes breathing, digestion, heart, and blood vessel function, swallowing and sneezing. The cerebellum controls motor movement, coordination, balance, equilibrium, and muscle tone. The midbrain and the hindbrain, which make up the brain stem, culminate in the spinal cord, whereas inside the cerebral cortex, the gray matter lies outside and white matter inside. In the spinal cord, this arrangement reverses as the gray matter resides inside and the white matter outside. Paired nerves exit the spinal cord, some closer in direction towards the back and others towards the front. The dorsal nerves receive sensory information from skin and muscles, and ventral nerves send signals to muscles and organs to respond. Studying the nervous system. The study of the nervous system involves anatomical and physiological techniques that have improved over the years in efficiency and caliber. Clearly, gross morphology of the nervous system requires an eye-level view of the brain and the spinal cord. However, to resolve minute components, optical and electron microscopic techniques are needed. Light microscopes and later electron microscopes have changed our understanding of the intricate connections that exist among nerve cells. For example, modern staining procedures make it possible to see selected neurons that are of one type or another or are affected by growth. With better resolution of the electron microscopes, fine structures like the synaptic cleft between the pre- and postsynaptic neurons can be studied in detail. Along with the neuroanatomical techniques, a number of other methodologies aid neuroscientists in studying the function and physiology of the nervous system. Early on, lesion studies in animals provided information about the function of the nervous system by ablating parts of the nervous system or using neurotoxins to destroy them and documenting the effect on behavior or mental processes. Later, more sophisticated microelectrode techniques were introduced, which led to recording from single neurons in the animal brains and investigating their physiological functions. Such studies led to formulating theories about how sensory and motor information are processed in the brain. To study many neurons, millions of them at a time, EEG techniques were introduced. These methods are used to study how large ensembles of neurons representing different parts of the nervous system with event-related potentials or without stimulation function together. In addition, many scanning techniques that visualize the brain in conjunction with methods mentioned above are used to understand the details of the structure and function of the brain. These include a CAT scan, which uses X-rays to capture many pictures of the brain and sandwiches them into 3D models to study it. The resolution of this method is inferior to MRI, which is yet another way to capture brain images using large magnets that bobble hydrogen nuclei in the brain. Although the resolution of MRI scans is much better than CAT scans, they do not provide any functional information about the brain. Positron emission tomography, or PET scans, involves the acquisition of physiologic images of the brain based on the detection of positrons. Radio-labeled isotopes of certain chemicals, such as analog of glucose, enters the active nerve cell and emits positrons, which are captured and mapped into scans. Such scans show how the brain and its many modules become active or not when energized with entering glucose analog. Disadvantages of PET scan include being invasive and rendering poor spatial resolution. The latter is why modern PET machines are coupled with CAT scanners to gain better resolution of the functioning brain. Finally, to avoid the invasiveness of PET, functional MRI techniques were developed. Brain images based on fMRI techniques visualize brain function by changes in the flow of fluids in brain areas that occur over time. These scans provide a wealth of functional information about the brain as the individual may engage in a task, which is why the last two methods of brain scanning are very popular among cognitive neuroscientists. Understanding the nervous system has been a long journey of inquiry, spanning several hundreds of years of meticulous studies carried out by some of the most creative and versatile investigators in the field of philosophy, evolution, biology, physiology, anatomy, neurology, neuroscience, cognitive science, and psychology. Despite our profound understanding of this organ, its mysteries continue to surprise us, and its intricacies make us marvel at this complex structure unmatched in the universe. Wow, that was a lot to take in again. But we're getting there. We're getting there. We're getting closer, and it's all very, very exciting. So I hope you enjoyed that chapter. And if you like the show, share it with someone you know and give me a thumbs up or subscribe. I want to help the universe live a more inspired life.